This is the first of several podcasts that deal with the GI system. In the first podcast, we're going to give you a brief overview of the GI system and then talk about the oral cavity. And then in the subsequent podcasts, we'll talk about the various organs in the GI tube. So for the GI tract, your learning objectives, you should be able to describe the general histological plan of the GI tract. Would like you to be able to describe abrupt histological changes that occur in the GI organs. You should be able to describe the unique features of each of the GI organs. You should be able to describe features of specialized cells within the GI organs, and you should be able to identify the GI organs on glass slides and on histological images. We'll start with the oral cavity, and the oral cavity has essentially three types of epithelium. The lining mucosa of the epithelium is a stratified squamous epithelium. You'll find this on the ventral surface of the tongue, lining the buccal cavity, in the vestibule, and on the soft palate. This epithelium overlies a lamina propria, which is a loose to a dense connective tissue, and there may be mucus glands associated with that connective tissue in the lamina propria, and this overlies skeletal muscle. There's also a masticatory epithelium, which is a keratinized stratified squamous epithelium. You find this on the gingiva and the hard palate. This will overlie a lamina propria of dense connective tissue, and under that dense connective tissue is usually bone. And then there's specialized mucosa in the oral cavity. It's a keratinized stratified squamous epithelium with papilla, and you usually find this on the dorsal surfaces of the tongue. There are some patches of this specialized mucosa in the soft palate and in the pharynx. And of course, the specialized papilla often involve taste buds. On the left, we show the hard palate. It's a stratified squamous epithelium overlying bone. And on the right, it just shows a gross anatomical view of the tongue, which is the largest structure in the oral cavity. The tongue, of course, is covered by a stratified squamous epithelium over connective tissue with glands and skeletal muscle. The specialized papillae in the tongue, which we'll look at in just a minute or two, would be the filiform papillae, the fungiform papillae, which have taste buds associated with them, circumvallate papillae, which have associated taste buds, and the foliate papillae, which also have associated taste buds. Taste receptors initiate cascades of chemical reactions within specialized sensory cells in the taste buds, and these often involve G-protein receptors or ion channels. As we've suggested, the papillae cover the dorsal surface of the tongue. The filiform papillae are the smallest and most numerous. They are not associated with any taste buds. The filiform papillae are distributed over the anterior surface of the tongue. They have a conical, elongated, keratinized, stratified squamous epithelium. And if any of you are familiar with cats and have been licked by your cat, you know the sandpaper feeling of the cat's tongue. That is primarily because of all the filiform papillae on the cat's tongue. Fungiform papillae, on the other hand, are large mushroom-shaped structures with taste buds. They're on the dorsal surface of the tongue in humans, and they're scattered among the filiform papillae. These are actually visible to the naked eye as small spots. And then circumvallate papillae are large, dome-shaped taste buds. They're on the anterior sulcus. There may be 8 to 12 circumvallate papillae in humans. The circumvallate papillae are surrounded by a trench into which enter a serous secretory product from von Ebner's glands. And then foliate papillae are parallel ridges on the lateral surface of the tongue. These also have taste buds associated with them. There are serous glands that empty into associated clefts with these taste buds. And interestingly, the foliate papillae are not always visible in aged individuals. This slide just shows a general histological overview of the 4 types of papillae. Here, the filiform papillae, these are very conical type structures covered by a stratified squamous epithelium. Foliate papillae shown here, a broad surface with taste buds along the lateral surfaces. There are some trenches. Minor salivary glands empty into these trenches. Here you can see a few ducts. You can notice some skeletal muscle here. Plenty of skeletal muscle here, of course. Some of the fungiform papillae shown here, and the very large circumvallate papillae with the very deep trenches, these serous glands of von Ebner that empty into these trenches. And the secretory product here washes taste stent molecules out from the trenches so the taste buds will sense new tastes. This is just a little bit higher power view histologically of the taste buds. There are 3 taste buds shown here. Here you can actually see a taste pore. You can see several different cell types within the taste buds. Again, a much higher histological view showing the taste pore and the diagrammatic view on the left side. The taste buds have several types of cells. There's a basal cell, which is a generative cell. You can see that here on the histological profile. There are 3 types of axons in the taste buds, type 1, type 2, and type 3 axons. These are all sensory cells. We believe there's a progression in the development of these axons in the taste buds. We're not going to ask you to distinguish the different types of axons. You can also see in the histological section some supporting cells. These are sometimes called sustentacular cells. Something that's interesting, all taste buds sense all 4 tastes, and complex taste sensation requires an intact olfactory apparatus, and the taste buds are relatively interesting because these basal cells are generative cells for the neurons. So the neurons and taste buds keep turning over, and that is very unusual. Normally neurons are in a G sub 0 stage of the cell cycle and they don't turn over, but the taste neurons do actually turn over. This cartoon just highlights something about the different taste sensations. So, for example, salt and sour sensation are involved with ion channels. Sweet and savory sensation have specific receptors, and bitter has a specific receptor, and these receptors for sweet, for savory, and for bitter are involved with G protein coupled receptors. And you learned a lot about G protein coupled receptors in your biochemistry course, so I don't need to go over them here. This image shows an overview of the plan of the GI tract, and we'll talk about this in quite a bit more detail, but generally the plan for the GI tract, once you get beyond the oral cavity, essentially it's a series of tubular organs, and we can define 3 major areas. Where I've got my arrow right now, that would be the lumen of this tube, and we can define a mucosa, which I'm highlighting like this, and the mucosa has multiple components that we'll talk about in detail: an epithelium, a lamina propria, a muscularis mucosae. Then there is a submucosa, a large layer of connective tissue. There's a muscularis externa, an inner and an outer layer of smooth muscle, usually smooth muscle, and then there is a serosal or an adventitial surface. That's not shown on this image, but we will talk about each of these components of the GI tract in more detail as we move through this podcast and the next podcast. This is just an overview of the GI organs beyond the oral cavity. Some things I'd like you to think about. The lumen of the alimentary canal, or of the GI tract, is physically and functionally external to the body. That's a little bit hard to wrap your mind around sometimes, but if you think about it, the luminal space in the GI tract is essentially equivalent to space outside the body. The alimentary mucosa is the surface across which most substances actually enter the body. So we take in food, that food goes into the lumen of the GI tract, but the food as it's broken down, the molecules don't get into the body unless they cross the mucosa. So this mucosa clearly performs many functions. It secretes digestive enzymes, acid, mucin, antibodies. It functions in absorption, so it absorbs the products of digestion as food is broken down. It absorbs, for example, vitamins, water, electrolytes, and lots of recyclable materials such as bile, salts, and cholesterol. The mucosa of the GI tract serves as a barrier against noxious substances, foreign antigens, and pathogens, and the mucosa in the GI tract performs a lot of immunologic functions. We've talked in other podcasts about gut-associated lymphatic tissue and mucosal-associated lymphatic tissue of the GI tract. There's abundant diffuse and nodular lymphatic tissue and many wandering T and B lymphocytes in the lamina propria and submucosa in the GI tract. Antigens are processed by macrophages and M cells. Plasma cells produce local IgA in the lamina propria and by various glandular secretions. Mast cells, which are connective tissue cells in the lamina propria and in the submucosa, can coordinate antibacterial activities of various white blood cells. Eosinophils within the lamina propria particularly are antiparasitic, and Paneth cells, which are part of the mucosal cells in the GI tract, can release antibacterial substances into the bases of the crypts in the GI tract, and we'll show you this in a little more detail later. The GI tube, to start with, consists of a mucosa, which has three components: an epithelium, a lamina propria, and a muscularis mucosae. And here I've drawn this to show the epithelium. Here I've just drawn them as flat cells. We'll talk about how this varies in just a minute. So there's an epithelium. The lamina propria I've just tried to draw to show things like maybe blood vessels, some connective tissue fibers, potentially some connective tissue cells, and the muscularis mucosae, and I've drawn this to represent two layers of muscle, an inner circular and an outer longitudinal layer of muscle, and we'll talk about each of these. The epithelium in the GI tract will vary depending on where you are in the GI tract. It'll be a stratified squamous epithelium in the esophagus. It will be a stratified squamous epithelium in the anus. Everywhere else, the epithelium in the GI tract is essentially a simple columnar epithelium. The lamina propria in the GI tract consists of a loose connective tissue. It's very rich in cells, lots of capillaries and lymphatic vessels. Portions of the mucosal glands will actually extend into the lamina propria. And then the muscularis mucosae consists of two layers of smooth muscle, always smooth muscle, and always an inner circular and an outer longitudinal layer of smooth muscle. So this is the mucosa. An epithelium, a lamina propria, and a muscularis mucosae. And here shows the epithelium on high magnification. This is the esophagus. It's a stratified squamous epithelium. Here would be the lumen, and then here is a little bit of the lamina propria. Here a little bit deeper in the GI tract, probably somewhere in the small intestine. I don't know with absolute certainty which organ, but I can see the simple columnar cells here for the epithelium, for the mucosa, and here would actually be a profile of the lumen. I think this will become clear later. Some lamina propria, connective tissue cells, and then muscularis mucosae. A nice example of that: inner circular layer of smooth muscle and outer longitudinal layer of smooth muscle. And then the submucosa showing connective tissue. Look at the blood vessels. Here's some profiles of blood vessels, probably two arterioles. This might be a little profile of a vein. This could be a profile of a lymphatic vessel. Here are some different connective tissue cells. So almost certainly these are fibroblasts. This cell might be a mast cell, for example. Moving on, we've got the mucosa. Then there's the submucosa. It's a connective tissue layer with blood vessels, lymphatics, maybe parasympathetic ganglion. Sometimes there are glands in the submucosa. And then there's a layer that's the muscularis externa. And I've drawn it this way, trying to highlight the inner circular and outer longitudinal layer, and it's usually smooth muscle, although it can vary a little bit depending on where we are in the GI tract. We'll talk about that. The muscularis externa is always an inner circular layer and an outer longitudinal layer, with the exception that in some portions of the stomach you pick up a third middle layer of smooth muscle that's a little bit more oblique. You'll see that in the laboratory. Then finally, there's a serosal layer or an adventitia. The serosa would be a surface that's covered with mesothelium, and it would be a surface that's free in the abdominal cavity, for example. If the organ is retroperitoneal, then it would have an adventitia, a connective tissue layer that would blend into the connective tissue of the other organs in the body. Here's an example of the small intestine, the mucosa, muscularis mucosae shown here, submucosa with some submucosal glands, so I know that this image is an image of the duodenum because of these submucosal glands. Here is the muscularis externa with an inner circular layer of smooth muscle and an outer longitudinal layer of smooth muscle, and some of the adventitia shown here. The image on the bottom is a high power view on the external region of the organ. So here we're seeing the mesothelial surface, the inner circular layer of the muscularis externa, the outer longitudinal layer of the muscularis externa, and look, here is a layer of nerve cells within the muscularis externa. This myenteric plexus, as it's called, is generally found between the inner circular and outer longitudinal layers of the smooth muscle. Look at the large neurons showing the nuclei and the nucleoli. Here is a view showing submucosa with a submucosal plexus shown here, also called a Meissner's plexus. Here is a little bit of muscularis mucosae, lamina propria, and the epithelium. So from here to here would be the mucosal surface with the epithelium. Here I'm in the lumen of the gland. And then down at the bottom of the field, I'm catching a little bit of the inner circular layer of the muscularis externa.